Now see, gynecium is the fourth pole, or it is the female reproductive part. Its unit is carpel, also called pistil. In a flower, if these pistils are more, if it is more, then this pistil may fuse with each other. This condition is called syncarpus. And when these pistils are free, not attached with each other, this is called apocarpus. In a single pistil, its different parts are pit A, stigma, onto which pollen grain lands or platform place for the pollen grain. Second part is the style. Through this part, the pollen tube extends, and next the swollen part of a pistil, or the basal part of the pistil which is swollen, it is called ovary. Inside the ovary, the cavity that is present is called ovarian cavity or locule. From the inner wall of the ovarian cavity, there is a special tissue that arises. This is called placenta, whose teeth contain ovule, also called megasporangium. Now see, in the ovary, from its inner layer, placenta arises. This is the diagram of placenta. The whose teeth part has got ovule or megasporangium. Detailed diagram of the ovule, or the enlarged view of the ovule is, this is the outermost layer of the ovule, that is, these two layers comprises of integuments, in which its outer layer is outer integument, next to it is inner integument. The outer layer is outer integument, next to it is inner integument. These two integument layer that encircle the ovule, not completely. At T part, this is a T part where the integument layer is absent. That T part is a micropyle. Next to the integument layer, this layer is called nucellus layer. All these cells are diploid. All these cells are diploid. The integument layer responsible for giving protection to its innermost layer. Nucellus layer also responsible for giving protection to it. In some seed, the nucellus layer usually persists, whereas in most of the seed, nucellus layer get destroyed. Below the nucellus layer, there is a special structure. This structure is called embryo sac. Structure is called embryo sac. It is also called female gametophyte. Female gametophyte. As we know, the male gametophyte is represented by pollen grain, and the female gametophyte is represented by this embryo sac. This embryo sac comprises of following structure. See here, towards the micropylar end, the three cells are present, out of which the two cells that are present at the T part are called synergid cell. From its cell wall, finger-like structure modification arises called filiform apparatus. This finger-like structure modification is called filiform apparatus, which releases chemical so as to attract the pollen tube towards itself. At the bottom of synergid cell, there is egg cell. Just opposite to the micropyle, there is a region that is called slaja. At the slaja end, the three cells that are present are called antipodal cells. In fact, antipodal cells do not carry out any function. The two synergy cells do not carry out any function. At the middle, there is a large cell that is called central cell, in which there are two nuclei, and these two nuclei are called bipolar nuclei. In some book, it is also written as polar nuclei. So, thus the embryo sac is a 7-cell structure, and towards the micropylar end there are 3 cells, towards the antipodal cells there are 3 cells, and at the center there is a single cell. Out of these 7 cells, the functional cells are central cell and egg cell. Remaining cells do not carry out any function. Now, about megasporogenesis. Before dealing to the megasporogenesis, let me recall the structure of a ovule. This is the structure of a mature ovule, in which its outermost wall is integument. That is outer integument, and this is inner integument. It encircles nucellus cells, that is given here. And within the nucellus cells, there is embryo sac that is present at the center. Let me recall the structure, how the embryo sac formation here occurs. Initially, these are the nucellus cells, and at its center, the cell that is present, that is called primary sporogenous cells, which form megaspore mother cell. And is represented by MMC. It undergo meiosis cell division, that is megasporogenesis. In fact, during megasporogenesis, from the megaspore mother cell, which undergo meiosis, it produces 4 haploid megaspores. This is definition of a megasporogenesis. So, megasporogenesis is defined as the formation of 4 haploid megaspores from the megaspore mother cells by meiotic cell division, which is given in the diagram in different phases. That is, at the end of meiosis 1, 2 haploid cells are produced, which are called dyads, and then at the end of meiosis 2, your 4 haploid cells are produced, which are called megaspores. After megasporogenesis, the megaspores that is present towards the micropylar end, mark the here micropylar end. Here it must be the micropylar end. The 3 megaspores that is present at the top degenerate, that is given here, and the lower one megaspore, that is the only functional megaspore towards this larger end, that is given here, towards this larger. This functional megaspore get enlarged and differentiated to form embryo sac. Now, how it forms embryo sac? See here. Initially, this megaspore contain haploid nucleus, in which its nucleus undergo 3 times continuous mitotic cell division. That is, 3 times continuous mitotic karyokinesis. Only first time initially, this nucleus undergo repeated continuously mitotic karyokinesis, which result in formation of 8 nucleus structure. Then cytokinesis occurs, in which the 3 nuclei that is present towards the micropylar end, this is here micropylar end, they get arranged to form 3 cells. These 3 cells are 2 are synergic cells located at the top. From its cell wall thickness, finger-like projection arises, which are called filiform apparatus, whose main function is to attract pollen tube towards itself. and then beside this the two synergid cells do not have any function. In between the two synergid cells at its bottom there is an egg cell which is a functional one. All these two synergid cells along with egg cell together constitute egg cell apparatus. At the opposite end of the micropylar end, that is at its larger end, the three cells that are present, these are antipodal cells, which are also a functional case. At the center part of the embryo sac, there is a large cell that is called central cell, which contains two haploid nuclei, that is polar nuclei. Remember, these two nuclei remain in separate condition and not in fused condition. Thus, a mature embryo sac is 7 cell structure. Here 1, here 1, here 1, here 1, 4, 5, 6, 7. Out of this 7 cell structure, only functional cells are egg cell and the central cell. And beside these two functional cells, other cells degenerate. This is, though it is a 7 cell structure, but comprised of 8 nucleus. Here 1, here 1, here 1, here 2. That is 5 nucleus. 6, 7, 8. Thus, the mature embryo sac is a 7 cell structure and 8 with 8 nucleated structure. This is all about megasporogenesis. See, let me differentiate microsporogenesis from megasporogenesis. During microsporogenesis, from one microspore mother cells, it divides meiotically to produce 4 microspores. Whereas during megasporogenesis, one megaspore mother cells divide to produce 4 megaspores. Remember, all these microspore and megaspores are haploid structure. All the microspores in this case are functional. Here, 3 megaspores degenerate and 1 functional. 4 microspore differentiate to form 4 polymeric or 4 male gametophyte. Here, only 1 megaspore differentiate to form embryo sac or female gametophyte. It takes place in microsporangium. It takes place in megasporangium.